Okay, again, good evening to everybody who joined us today. Today we have with us Professor Barry Bradford from Michigan State University. Bradford completed his dual bachelor degree at Iowa State University and a doctorate in animal nutrition at Michigan State University. He served on the faculty at Kansas State University from 2006 to 2019. In 2020, he returned to Michigan State University, and his research focuses on dairy cattle nutrition and metabolism, with particular emphasis on attempting to translate novel findings in fundamental metabolic physiology to practical application in animal agriculture. Contribution by Bradford Group have largely focused on dietary utilization of byproducts in lactation diets, the physiological impact of systemic postpartum inflammation, and the rules of nutrients as signals. He worked closely with 25 graduate students and postdoctoral scholars, as well as dozens of collaborators. Bradford has contributed to more than 100 refereed publications and shared those findings in more than 150 invited presentations around the world. We are very happy to have him here with us. And Barry, we'll let you have the stage. You can share the screen.
Okay. Great. Can you see my slide?
Yes.
Perfect. Well, thank you very much for this opportunity. I've enjoyed collaborating with Maya for a few years and got to meet Uzi here in Michigan a few months ago, and just really am honored by this invitation to share some ideas with you. And I hope we can have some discussion and share some ideas back with me. But if I'm speaking too quickly or not clear, or if you have any questions during the presentation, please just stop me while we're on a topic and let me know what your question is or what the problem is. With that, let me dive in. What I thought we could cover today was, first of all, the evidence behind this focus on transition cow inflammation. So I'll talk a little bit about just setting some baseline about disease in the transition period from a US perspective, and perhaps we can dialogue on how it's different there or not. And then talk a little bit about what we understand in terms of the physiology of inflammation at the time, maybe some of the reasons why this isn't necessarily completely bad, but then also talk about the consequences of too much inflammation, and then talk mostly about how we can take this to farms. What can we do to assess whether this is a problem on a given farm, and what are some strategies for influencing inflammation in transition cows? So first of all, I use this data set a lot. It's from Denmark. I don't think it's really any different than what we would see in the United States, but in this data set from commercial farms in Denmark, they simply plotted out when diagnosed diseases occurred relative to the day of calving. And this is given as a proportion of the total population. So one way to think about it is cows that are between a month and 3 months into lactation have about a 1 in 1,000 risk of having some kind of diagnosed illness in the day. So 1 out of 1,000 cows is sick in some way in a given day. But in the 3 or 4 or 5 days right after calving, that number jumps all the way up to almost 1 in 10, regardless to some extent of whether they're first lactation or older cows. So we see this massive jump in disease risk. And actually, if you add up these diagnosed diseases in the first 2 weeks of lactation, for most farms that I've looked at their data, that makes up over half of the total disease on the farm. In mature cows, if you ignore the calves. Okay, so immediately, after being born, calves have a very high risk, and then right after giving birth, cows also have a very high risk. So if we can do something to reduce the risk here, we have a huge impact on total disease incidence on the farm. And that's one reason why we really focus on this period. What makes it challenging, though, is it's not like this is just one pathogen, we just need to get rid of a pathogen, or it's just one condition to fix. It's a big mixture of problems, right? And maybe on average, it's maybe 40% metabolic disease, 60% infectious, but that doesn't really show the fact that many of the cows that have major problems, it's a mixture of things going on. Maybe she started with a metabolic problem and then she also got metritis, for example. And so these make them very difficult problems to solve. I'm showing here, it's a little bit old, but the newest national data we have published for the US is from 2014, and actually a survey from a little before that. So it's probably 10 years old. But in that data set, this is producer reported incidence of disease on dairy farms with hundreds of farms surveyed. And you can kind of see the range of problems that we deal with in transition cows. I didn't include mastitis in here. Of course, we have more mastitis in transition period, but that's not only the transition period. So if we look at these mostly transition problems, we have roughly 5% of cows that have some kind of calving issues, 4 or 5% with reported ketosis, although that number is probably much higher if we wanted to look for it carefully. Retained placenta, that's still probably a 4 or 5% condition. And metritis is another one where farms report 7%. If people look very carefully, if researchers look very carefully for it, often it's more like 30 or 40% of cows with metritis. And perhaps that's what you guys see, because I understand that veterinarians are looking at every single fresh cow, which is not the case in the United States. So it would be interesting to compare those numbers, and it's probably a lot of the difference would have to be due to diagnosis as opposed to true differences in health. Displaced abomasum and milk fever are two conditions that we have had success, and I think in Israel as well, in the last 20 or 30 years of reducing the incidence of those with nutritional strategies, but they remain a problem in some cows. And so typically studies that look carefully at all the cows in a research type approach, we use roughly 40 or 50% of cows through the transition period have something that you can pick up that's a problem, at least subclinical ketosis or mild metritis for some time. So it's a very common problem. Okay. One thing that I like to point out to dairy producers here when I talk about this is they'll often look at these diagnosed clinical issues where really they're only reporting cows that have to be treated. And of course, different farms differ widely in how aggressive they are with treatment. I know farms that still treat almost every single cow for ketosis just as a preventative issue, whereas others don't even look for problems in hardly any transition cows. So when they look at their disease rates, their key performance indicators, their KPIs will include, you know, how much displaced abomasum might be acceptable. Oh, I don't know where that green line came from. Sorry. But anyway, they'll look at those numbers and they might say, we have an acceptably low rate of disease, it's not a problem. And they'll say, you know, most of these cows recover, they don't have to be cold, they don't die. But I like to point out to them, it's very hidden, but we have some substantial long-term costs from these diseases, even if cows recover. So these are all studies where researchers went to commercial farms and followed cows that had some disease after calving, compared to herd mates in the same pens that didn't have disease, and just asked how much did their productivity differ over the whole lactation. And again, what they found is rather surprising. About 500 liters reduced milk yield from mastitis in the first 2 weeks post-calving. Over 350 liters less milk from metritis, and ketosis about over 300 liters less milk. And again, these are all in the cows that recovered. And so most of that is coming from maybe making half a liter less for many months, and it's difficult to pick up. Unless you do careful statistical analysis of hundreds of cows, you couldn't find it, but it's a true cost to the farm, right? Probably an even bigger cost is, again, if you track those cows and ask, did they actually stay in the herd until they had another calf and stay in additional lactations? Again, maybe they weren't culled next week after the disease, but if you track them and see how poor they do in terms of reproductive efficiency, and maybe they were AI'd 4 times and then they were marked as do not breed and they ended up being culled at the end of lactation. We can track that from these diseases. So if we look at mastitis or ketosis, we have cows with more than a 3-fold increased risk of leaving the herd in that lactation compared to their healthy herd mates. In metritis, it's about a 1.5-fold risk, but metritis is a more common condition, than mastitis. So that's affecting a lot of cows as well. Okay, so anyway, these are very expensive long-term problems. Let me see if I can get rid of this mark. I don't know what's going on. Maybe it doesn't bother anybody, but it bothers me.
I think you should unshare and share because somebody did with the iPad.
Oh, okay. It wasn't actually me. Okay, I can do that. That's why I could only see it on one screen, I guess. Ah, there we go. Thank you. That's why Maya's a good collaborator. Okay. Get my laser pointer back. Okay, so many of you are very familiar with the concept of inflammation, but just to make sure we're all on the same page, inflammation is an important component of activating the immune system. Now, maybe we're used to thinking this again as a negative thing. It's associated with pain, fever, swelling, redness, all the things that we don't like when we're sick, right? But it is absolutely necessary. And I'm kind of mean to my kids when they get a fever. They have to be in pretty good pain before I give them anti-inflammatory drugs, because I've read too many studies about mice dying from getting too many anti-inflammatory drugs during a true illness. So if we're really fighting a pathogen, this is an important part of activating the immune system. Now, in an ideal situation, what's really interesting about the immune system is if you pull a white blood cell out of your body and stimulate it with something from a bacteria, it will, even that one cell on its own will start making inflammatory molecules. It will start activating, it will start this response. And interestingly, 3 or 4 hours, even if you don't change what you're putting on the cell, it will change to this resolution phase. It will start making anti-inflammatory compounds to start to calm things back down, to bring it back to normal. So it's a pre-programmed, this green line is a pre-programmed response curve that the immune system ideally will go through. And, of course, the logic is during that inflammation stage, you kill the pathogen and then you get the body back to normal, right? Because if we stay in that state of inflammation long-term, it's not good for other systems of the body. But that's kind of what the red line shows. This can happen if you have repeated insults, like we might have in some transition cows, or if you have the system failing to resolve for whatever reason. Uzi might say a lack of omega-3 fatty acids, right, available in the system to help with resolution. We can end up with this chronic inflammation, and of course, lots of human conditions, we can recognize that if it's arthritis or, there's lots of examples of that. All right. So again, this initial wave of inflammation can be triggered by pathogens. That's normally what we think about, but also tissue trauma. And I think after giving birth, that's probably a component of this in cows, right? Particularly with dystocia and stuff. So it's important to recognize that ruptured cells release cell contents that actually trigger inflammation as well. Okay, so then we have this question of when we're talking about inflammation, what are we talking about? Are we talking about a short-term resolved inflammation or a chronic one? And I think it makes a big difference in terms of what are the real impacts of that. So we'll spend some time on that. Another little bit of the physiology that I have to introduce before I show you data is one component of this whole body inflammation response is called the acute phase response. And this is simply a system where the liver actually plays a role in the immune response to an infection, because when it sees these immune system signals called cytokines, it actually starts making proteins that are released in the blood in very high concentrations, at least if it's a serious infection. And they help to do a number of things, like some of them will bind minerals so that bacteria can't have the minerals to grow with. Others will sort of combat oxidative stress, which can make the immune system more challenged to work properly. And so they have different roles. We don't totally understand what all of them even do, but I wanted to introduce this because we actually use these as biomarkers as well. So beyond their function, we can at least assess them and see, is something going on inside the animal that maybe we can't see through fever or sickness behavior, but maybe they still have some of these signals going through the system that's changing them internally, and we can only pick them up by measuring something. And one of the reasons we don't measure these primary signals is they disappear very quickly. So we could have a very severe inflammatory challenge, and these might jump by 10,000-fold and then come back to normal 5 hours later. And so if you didn't pull the blood sample at the right time, you may not even see any change. Whereas if we look at these proteins made by the liver, they have a much longer life, and so they'll stay elevated for 2 or 3 days often. And so it gives us a better chance to find what's been going on in that cow recently. So here's an example of a data set using that kind of approach. This is from work from Cornell, an observational study. So they're just taking 70 cows, taking blood samples once a week before calving and much more frequently after calving. And in this graph here, they're looking at haptoglobin, one of those acute phase proteins. So we can use this as a tool to say, does this cow have some, what I would call subclinical inflammation? Again, her ears may not be droopy, she may not have an obvious fever, but she still may have something going on. So in this data set, once they had the data on haptoglobin, they said, okay, now we're going to break this into groupings and look at the 25% of cows with the highest haptoglobin after calving. So that'd be this group. And then the middle upper quartile is the third quartile, second, and then first. And so in this group of cows, maybe a healthy group compared to some that have been published, basically half of the cows don't show a noticeable jump in this biomarker after calving. So they don't show obvious signs of systemic or whole body inflammation based on this marker. But the other half of the cows, what we can see here is somewhere between a 3 to 5-fold increase in this marker. So again, just to bring it back to just normal, how would we interpret this in medicine? If you went to the doctor and said you hadn't been feeling well and they did a big blood panel on you and looked for a bunch of things, if they saw a 5-fold jump from normal in this acute phase protein, they would want to do follow-up tests, right? Something is going on. The negative part about these markers is they're not very specific. It could be that you have hepatitis, you have a liver infection. It could be that you have heart disease. You know, there's a lot of things that can feed into this, and we'll talk about that in cows. So they're not very specific, but what it does tell you is something is a little out of whack. This isn't a normal thing you expect to see in a healthy animal, okay? Now, people working in dairy cows on health and physiology have for decades now talked about a transition period, which we usually define as starting about 3 weeks before calving and continuing through 3 weeks after calving. Now, this study didn't track before 3 weeks before calving, so we can't really look at changes out to there. But you can see that there's a lot of sort of bouncing around going on in this marker right up to 21 days after calving, and then things seem to be pretty steady after that. Okay, and that's really why this window was defined, is people observed over many years of observing and studying that that's the window when things are kind of moving around a lot, that the cow is going through a lot of physiological changes. And I just throw this in there because I think it's such an interesting finding. So this was published last year from a study of women. And the core question of this study was, can we look at a whole host of different markers in blood and predict when a woman will actually give birth? Okay, so they would like to say, we'll look at a blood marker and say, oh, you're actually going to be a week late in giving birth or 2 weeks early or what have you. And what they found was, particularly by looking at changes in markers of the immune system and in general hormonal signaling, what did they say? That we start a transition period 23 days before labor, okay? Just almost identical to what we see, what we've sort of crudely defined in cows as that transition period. Again, 9-month gestations in both. So it's just fascinating. There's probably a lot of this that we've spent so much time working on in cows. I mean, my bias would be that we're kind of leading the understanding of this kind of system in mammals, in what we're working on in cows. And I would say, I do like to point out, I've had people ask, I think it's a reasonable question, when we show these inflammation signs in dairy cows after giving birth, are we finding something that we've kind of selected cows accidentally that have all these health problems after calving because they make too much milk too quickly? And some of that may be the case, but what I will say is in other species that have been studied, we do see this inflammation scenario after giving birth, even if they're not making nearly as much milk as a dairy cow. Okay, so I don't really think it's a dairy cow specific situation. Okay, so if you buy that, or let's for a moment assume that that's true, that we should expect some inflammation after giving birth in almost any dam. If that's the case, you might think then that maybe it's there for a reason. Maybe it's not just an accident. Maybe there's something beneficial about this. And let's explore that for a second. There's a study that was done in Canada a few years ago now, where they got some very clear data that helps us sort of peer into this physiology a little bit. So the question they asked was, is it maybe the case that cows after calving don't eat as much as we would like them to eat simply because they're in pain? Maybe they're not feeling good. They don't want to stand up and walk to the feed bunk. And if we can relieve that pain, maybe they will feel better and go start eating, and then we can avoid some of the problems with displaced abomasum and ketosis. Seems like a good hypothesis to me. So they said, well, we can test this by using an anti-inflammatory drug. We know it is banamine here. Flunixin-meglumine is the compound in banamine. And they gave this treatment 2 hours after calving and again 24 hours after calving. And this group does an excellent job of doing large studies. So they actually put over 1,300 cows on the study. Unfortunately, it wasn't such a positive outcome as they had hoped for. So in this graph, I'm showing odds ratio. All that means is if we set the rate of a problem in controls to 1, the treatment group in the blue bar shows you how prevalent that problem was in the treatment group. So for retained placenta, the cows that received the anti-inflammatory drug had 2.5-fold greater risk of having a retained placenta. Okay, so much, much more. And again, this is hundreds and hundreds of cows on the study. Probably because of their retained placenta, they also had a 50% increase in their risk of metritis. So it did not go well for this treatment. Why is that? Well, it's easy to say after the fact, but when you see these results and start to think about what might be going on, there's actually quite a bit of literature in the human side in human medicine documenting that use of anti-inflammatory agents around the process of giving birth can actually disrupt the normal process, the birthing process. And I'll show you some data on that in a second. But the other thing I want to point out briefly in this study, if you read the paper carefully, they talk about how their first protocol for this study was to give the anti-inflammatory drug 24 hours before expected calving, and then I think again right after. Well, they started on that protocol and maybe they enrolled 300 cows, and they did some early statistics and they had to stop that treatment because they had already shown a significant increase in stillbirth. So actually the cows that got treated with banamine a day before expected calving had over a 12% incidence of stillbirth. So it was something like a 5-fold increase compared to controls. So even more dramatic, if you stop the inflammatory waves before the calving process has begun, it's very problematic. So again, if we go back and dig through the human literature, we actually know some pretty neat mechanisms now that explain why inflammation might be very important for parturition. One of the things that happens is as the fetus is nearing, parturition, it will start releasing signals that will actually help to attract immune cells to the uterus and to the cervix. So we have different types of white blood cells coming in through the bloodstream, entering the endometrium and the cervix. And one of the things that those cells do is they produce proteases, enzymes that break down the connective tissue so that the cervix can start to loosen and ripen. Okay, so that obviously is essential for allowing parturition to occur. The other piece is that these same inflammatory signals will actually help drive prostaglandin production, and prostaglandin will work on the smooth muscles to cause contractions. And so if you think about the retained placentas, even after a calf had been born, those muscle contractions need to continue to release the placenta, right? And if we use an anti-inflammatory drug to block the inflammatory signals that cause muscle contractions, they stop, and we stop the process, of getting rid of the placenta. And so it sort of makes a lot of sense. And actually, then if we think about, well, why do we have a lot of cows that have at least modest signs of inflammation after giving birth? This is probably part of it because, yeah, the signals are concentrated around the reproductive tract, but there's no real mechanism in the body to completely prevent them from escaping back into the bloodstream. So we're going to have some of these signals going to the rest of the body, having at least some impact on other organ systems. Okay. So if I have to guess, and I really can't prove it, but if I have to guess, the healthy cows that we see little small increases in inflammation after calving, I think we can probably attribute to simply that process of parturition, the inflammation needed for that to occur, and to some extent the tissue trauma that comes from that process probably contributes. But that doesn't necessarily explain why we have a wide range of responses across cows. So some cows, again, have tenfold greater markers of inflammation than others. And I think there we have to think about other things that contribute. So certainly, there's lots of studies showing that if a cow has mastitis or metritis, again, the inflammation starts in that organ, but it's going to escape to the whole body. And so we can anticipate that those infectious diseases in a fresh cow are going to contribute to raising the level of inflammation in the whole body. We can also speculate about other things that might contribute. There's a ton of interest right now in whether transition cows have a leaky gut, whether there's bacterial components escaping the gut and entering the bloodstream, which goes right to the liver. That could contribute to inflammation. We don't really know. Even oxidative stress, if we don't provide enough antioxidants, one strange aspect of physiology is if you have oxidative stress, that promotes inflammation as well. So that could contribute. And we even have evidence of social stress. So the example I tend to give is there's studies, well, the beef industry in the United States moves cattle very long distances because of access to grass versus grain in feedlots. And so it's a continual problem of how do we deal with the stress associated with long distance trucking. And so some studies have been done where they just loaded up cattle in trailers and drove them around for 8 hours and then put them back in the same pen. And they show even that will cause a 2 to 4-fold rise in the acute phase proteins, these markers of inflammation. So, you could think about, and it's a speculation, but, is it the case that say a new heifer that has never been through a milking parlor before, going through a parlor, changing pens after she calves, do those kinds of stressors contribute as well? We don't really know. But there's a lot of, again, these are non-specific markers. They are altered by many things. And I think this is a great example of integrative physiology, where we have a lot of things that probably contribute to these markers that we're measuring. And I know Mayo's been working on some data related to this from heat stress, and we don't really know the mechanism there, but there's a number of ways that that could contribute as well. Okay, so ideally, if we want to take this to farms, what we would like to be able to do, just like a hospital would do in human medicine, we'd like to say there's some level that is acceptable, and then above that level, we're going to decide, okay, then it's not acceptable anymore, right? And that's difficult to do. That's not really real biology, but you have to come up with sort of yes/no answers. Is this a problem to deal with, or is it not? And so there have been probably 5 or 6 studies now that have used the acute phase protein haptoglobin to try to diagnose, to try to come up with a cut point to say, here's a level where we have more problems. And the way they've done that is they've tracked cows for a whole lactation and asked, okay, if we look at this hundreds of cows, if we draw a line here, will that give us the best differentiation between cows that make a lot of milk or that suffer in milk production, or cows that have a better odds of getting pregnant or not, and those kind of different outcomes. So in different studies with different outcomes, we end up with different cut points. But the larger studies that are done well, if I had to average them, we end up coming up with a cut point something close to 0.5 grams per liter. Okay, so I'm just going to use that now as an example. And if we look at cows in the first couple of days after calving, if we have cows that stay below that, that's kind of our reference point. Those cows give us what we generally consider to be successful transitions. They hit a high peak milk. They become pregnant at a relatively fast rate, have a low risk of leaving the herd due to disease. However, about half of all cows in the studies that have been done recently do cross that line at some point. Okay, so we're talking about a major proportion of cows. And importantly, that's not necessarily all the same cows that we could have identified by tracking clinical signs. So not all of those cows have illness behavior or high ketones or fever or anything like that. So about 25% of the clinically healthy cows will be above that line too. So we really are identifying cows that we can't identify just by a physical examination, all right? So we've drawn this line in the sand. Half of the cows are above, half are below. And what we find in these studies is substantially more risk of metritis. So some of that is because the metritis is probably part of what's driving the inflammation that we're measuring, okay? But this is picking it up before you can easily diagnose it. These cows are making 5 pounds per day less milk, or more, around 2.5 liters per day less milk, so pretty substantial there, and about a 20% drop in the pregnancy rate. So, if that's half of the cows, that's a pretty big impact on the overall herd, and obviously it would vary by herd, but across these studies, that's been kind of the take-home message, all right? So that's compelling, and you show data like that, and you have herds that say, well, or veterinarians that ask, well, what can we do? Can we actually identify those cows and do something about it? Unfortunately, today, that's not so easy. Maybe you guys have a system in Israel where this could work, but in the US today, the reality is, if you want to know if this cow is above or below this line, maybe we could sample 3 times a week, and maybe so on average you're getting cows at day 2 of lactation, and you send it to a diagnostic lab. So in the United States today, about your only option to get this done would be with a state level veterinary diagnostic lab. So you can probably get that set in 1 day, but then typically today at least, it's going to take you another 2 or 3 days to get data back from that lab. And now this cow is at least at 5 days in milk, probably more like 6 or 7 on average. And the problem is we have no data on, we only have 1 study where people have treated cows between days 6 and 10. And that 1 study was only 40 cows. So it has some promising results, and maybe that's a solution, but we don't really know yet. We don't have any evidence base to say, oh, you should do this. because there's been very little work done on that. So that really is a problem that we're trying to start working through. Several research groups are kind of working on that. But again, to take this to farms today, it's hard for me to tell them to spend quite a bit of money on these diagnostic tests when we don't know exactly what to tell them in terms of how to treat that cow if she is above that line.
In that cow, in that study, how did they treat the cows that were diagnosed?
If I remember right, they gave meloxicam. It's Stephen LeBlanc's group, and I can send you that paper. But yeah, they saw some metabolic benefits, but it was only 20 cows per treatment, so there's only so much you can find. Anyway, based on what we have available today, and honestly, based on the cost of these tests, we and some other groups are working on, can we get to the point where we can show that herd level monitoring is actually worth the time and effort? And so we're trying to start a big study now where we're going to enroll between 40 and 50 farms, probably 3,000 to 4,000 cows. And this is the protocol we're going to use. We're going to go to a farm, go to the fresh cow pen, and if it's a big enough farm, sample 20 cows of a range of parities. Any cow that's less than 7 days in milk. So we'll have a range of samples between 1 and 7 days in milk. And the question is, if we do that and we can draw, again, some cut point for the herd, do we actually see differences between cow herds with more inflammation versus those with less? Okay, and so I want to share, again, the Cornell group led by Tom Overton. He had a grad student that has just kind of finished up some work on this. And this is, I think, the first work where they've tried to ask this question. And they ended up saying that statistically, their best cut point was to say, if you have more than 10% of cows that exceed 1.5 grams per liter plasma haptoglobin, then you're considered a problem herd, okay? And their basis for that, their justification was if they drew that cut point, they showed a statistically meaningful 2.5 unit lower 21-day pregnancy rate, okay? So again, on an individual cow, that's maybe not that amazing, but if you think about a whole herd shifting by 2.5 units, that's somewhat costly, right? You're gonna lose days in milk in the herd. It's gonna cost you delaying pregnancy in this, okay? So this is some work that we need larger data sets, and some collaborators are working here with us on trying to develop a new data set to sort of work with Tom and try to come up with clearer answers here. But I think there's some promise that this kind of strategy might make sense. But we still need to, eventually I'd like to get to the point where we can say, I can show you data that if we treat this top herd with some anti-inflammatory dietary strategy, they actually improve a lot, whereas these bottom herds maybe don't, it doesn't make a big difference. We don't know that right now. And we'd like to get to the point where we can have that kind of clarity. Okay, so what our group has really tried to do in my research program is try to ask cause and effect questions. So rather than just observing cows and showing relationships, we wanted to ask, what if we add inflammation? Or what if we subtract inflammation? Does that move cows in a more positive or negative direction? We still haven't tried this strategy. I don't think it would work, but maybe someday we'll get to that. No, I'm just kidding. So, you know, we've had interest in individual cow treatments, partly to do clean experiments, and I'll talk about one example of that. But, you know, I'm ultimately a nutritionist, nutritional physiology, so I really would like to get to the point where we can use dietary strategies to make progress on this. So I'll talk about that a little bit too. Okay, so this is a study we published 6 years ago now. We had seen some exciting initial work on a research university farm, and we wanted to really test it in a larger study without spending 2 years to run the study. So we took this to a commercial farm of about 2,000 cows, and we randomized fresh cows to receive sodium salicylate, which is a very ancient anti-inflammatory drug, meloxicam, which is a much more modern anti-inflammatory drug, or a control placebo treatment. And because we knew of the work on retained placentas and the risk there, we wanted to avoid any risk, and we started treatments on average 24 hours after calving. And we didn't, any cow that had a retained placenta, when we would have treated her, we did not enroll her. Okay, so we didn't have that complication. We put 51 cows on treatment, and we did not include primiparous cows in this study, which I can talk about that later if you'd like. So this was the really exciting finding from this study. Okay, so again, we treated cows just at the beginning of lactation, and for the sodium salicylate treatment, over the entire lactation, all 305 days, we increased milk yield by 11%, so over 1,000 liters of milk. And that's, again, just from treating them 3 times, so 24 hours, 48 hours, and 72 hours post-calving on average, and a significant increase in milk yield, even out 300 days later. And for meloxicam, we saw a very similar response. Again, about 1,000 liters of milk, roughly. And the nice thing about meloxicam is the dose we gave was about 1/1000th of the dose of salicylate, just because it's a far more potent drug and it stays in the cow much longer. So we just gave a single bolus about this big to these cows, and that gave us 1,000 liters of milk. So pretty exciting. The downside, and I'll come back to this in a minute, and the most difficult part of running this study is we had to have enough people there to be in the milking parlor every 6 hours, because this farm milked fresh cows 4 times a day, and they had to be there to find every cow that was on the study and dump her milk for 2 weeks, because meloxicam stays in the bloodstream for a long, long time, and we couldn't allow any residues to be in milk. Okay, so that's some of the challenge of doing this, the drug approaches. Okay, the other question that we wanted to answer is, some people, might say, okay, you're getting another 1,000 liters of milk out of this animal that's already making a massive quantity of milk. Aren't you just going to make her sick from, are you going to starve her, or put her in really bad metabolic health? So our other big question was just simply, did she stay in the herd? So we blinded the farm to treatments. They didn't know what cows were on what treatments, so that it wouldn't bias their decision making. And this is simply a survival curve. So if they didn't survive, that doesn't mean they died. It just means they either were cold or they died. Okay. So the black line here is the control group, and every time this line drops, that's a cow leaving the herd one way or another. And over 365 days, about 40% of those cows left the herd, which is a pretty typical rate for a U.S. dairy herd for multiparous cows, right? The salicylate group is in the blue here. Statistically no different than control, although maybe they started to stay in the herd better about mid-lactation. But the meloxicam group in red, we saw a significant decrease in transition problems as a code for culling. That's how this herd put in as a reason for culling in early lactation. So they did stay in the herd better in the first 100 days. And again, by the end of that year, it was marginally significant, where they had dropped from a 40% removal rate to just 25% removal rate in that group. So this is really the only study that I'm aware of with clear evidence of better retention in the herd. I hesitate to say health. It's a little complex to say that's truly better health, but at least the farm saw less reasons to cull these cows. So that's exciting as well. I do want to point out briefly, other teams have done similar studies now. This study was done by Turner Schwartz, who now is working as a postdoc in my group, but he was at another university when he did this work, and they asked whether giving meloxicam, again, the same anti-inflammatory drug we tested, either 24 hours before calving or 24 hours after calving would improve the transition period, I guess. And they were especially interested in whether it would help cows that had calving difficulty. So a little bit surprisingly, they saw no benefit for milk yield or really anything else in cows that had calving problems. But the cows that had normal calving that didn't need assistance had as much as an 18% increase in peak milk yield when they got the meloxicam before calving. Okay, and that also brings up another point that I want to make clear. I showed you a study with banamine or flunixin where it clearly causes problems with the process of giving birth. But there are two very large studies now showing that meloxicam does not cause the same problems. So we have good evidence that it depends on the exact drug and the exact mode of action whether it's going to be an issue for the parturition process. And we don't have to throw out all of these strategies because of that concern. Okay. So again, as I noted, we've used drugs in part because we wanted to do very clean research. We wanted to say, this compound, we know it works a certain way, at least we're pretty sure it works a certain way. And if we give a treatment, then we can more clearly interpret how it had those effects. But longer term, partly for regulatory reasons, partly for practicality reasons and logistics, of course, it would be very nice to do the same type of thing through a feed component. And what's nice about this, and a lot of what I've focused on too, is this idea that now nutrients work in ways that we wouldn't have thought of 30 years ago. Okay, and this really exploded after the publication of the human genome, because they found in that genome there was like 300 genes that encoded for cell receptors, like these two shown here. They're G protein coupled receptors, and several of them were well understood, through the 1980s and 90s, but they had no idea there were so many in the genome. And a whole bunch of labs around the world started trying to understand, well, what are these G protein receptors in the human genome that we didn't know about? And many of them now have been described, and a huge majority of them are actually nutrient receptors. So we in cows make these sensors, if you will, on the cell surface that will bind to fatty acids, sugars, amino acids, even minerals, and they can change the function of cells. So we can even have nutrients that maybe aren't even absorbed by the gut, and they can be changing animal or human physiology if they're sensed by a cell lining the gut, right? So it really kind of changes the way we think about nutrients working in the body and opens up a lot of other possibilities. Okay. Now, some of these have been studied since the 1990s, so now we're kind of used to thinking about it this way, but the number of nutrients that can act in this way has really exploded in the last 15 years or so. And again, one that's Uzi and others in Israel have contributed nicely to, and I think have a really nice story behind them, are the omega-3 fatty acids. This is a study where they actually did a dose response in transition cows at the University of Florida. So all the cows received 1.4% dietary calcium salts or calcium soaps of fatty acids, and they just changed out which fatty acids were in these feed products. So the starting point was really probably a 6-to-1 ratio of omega-6 to omega-3 fatty acids. And then they basically replaced some of the omega-6 with omega-3. So as that number drops to 5-to-1 and 4-to-1, you're getting relatively more omega-3s compared to the R6 treatment, okay? So then if you look at how cows responded to these treatments, they started at 14 days in milk, so they actually didn't hit the transition window. They waited till cows were through that and then started this. It's pretty shocking how quickly the milk response shows up. Going from a 6-to-1 down to a 4-to-1 ratio, you can see that there's about a 5 liter per day jump in milk yield that's maintained through this 15-week feeding period. And importantly, that didn't occur just by cows losing body weight and putting that body fat into milk. They were actually eating more, and they had very similar energy balance. So the more heavy milking cows on the omega-3s had slightly more negative energy balance, but it didn't have a major effect. So they returned to energy neutrality within a week of each other, and really didn't have any significant change in body condition score. The same group had earlier done a study that wasn't a dose response, but they asked about timing of omega-6 and omega-3 fatty acid supplements, again, with a very similar feeding rate. The omega-3 fatty acids here were 1.5% of diet dry matter as calcium salts of fish oil. And so the omega-3s were only fed around the time of breeding. Okay, so they tried feeding either saturated fat or omega-6s right after calving, and then they went to a saturated fat or omega-3s during the breeding period. And what they found is feeding omega-3s during the breeding period significantly reduced pregnancy loss. So if you average these two numbers, they looked at about a 12% pregnancy loss, and that's between the first pregnancy detection and the recheck. Okay, so between 32 and 60 days post AI, and it dropped to about half, to about 6% pregnancy loss during that window if they were getting omega-3s at the time. So there's potential benefit. The way I think about it is in terms of metabolic health early after calving, and I think that's reflected to some extent in just straight up milk yield in the previous study, but also potentially some benefit by making the system more anti-inflammatory, which we believe should help with pregnancy retention, improving, my brain just stopped, how the uterus sort of latches on to that fertilized embryo, after AI, so that you get a pregnancy that's actually going to last. Okay, so I think there's pretty exciting applications. Now, there's some difficulties with omega-3s. They're very expensive products. You can feed different things. Some of them are less expensive than fish oil, but it's of course tricky. You have to do some interesting things feed-wise to actually get those through the rumen, right? So we have to either use calcium salt, or we have to process flax or something like that, to try to do these things. So there's also interest in other approaches, and one that we've gotten involved in is, you can call it lots of things. You could call any of these compounds that we're talking about nutraceuticals. Some people use that term, basically using nutrients as if they're drugs. You could think of it as Eastern medicine. And in fact, Chinese skullcap is an example of a herb that's been used by the Chinese for many centuries, probably thousands of years. And really just in the last 20 years, there's been a massive amount of research with these kinds of compounds, including Chinese skullcap, in mice and cell culture, showing that at least if you provide enough of these compounds, they can potentially have anti-cancer effects, potentially anti-liver disease, anti-obesity in some cases, and anti-inflammatory effects. But we got involved after doing some of our drug studies and connected with a company in France that had developed a product and was actually selling it in the market, but didn't have what I would consider to be robust data in dairy cattle. And so we teamed up with them. We said we thought we could do a valid study with decent numbers to try to test whether some benefits of this could be shown in dairy cattle. So here again, we went to a commercial farm to try to achieve the kinds of numbers we needed in a reasonable timeline. So we ended up putting 40, again, multiparous cows on treatment for each treatment in a randomized block design. And to achieve that and give individual treatments, we used Lely robots to, recognize an individual cow, determine whether she should get pellet A or pellet B, and again, give individual cows her treatment in that way. And this is what we found, again, to try to mimic what we saw in the drug studies. I'm just going to mostly focus on milk yield here. So we fed this Scutellaria bicalensis extract for 60 days in this green treatment here. And 60 days is shown in this dotted line. So during the treatment period, we saw a tendency for an increase in milk yield. And of course, it didn't really show up till about week 3 that we started to see some noticeable responses. But then when we looked at the carryover period, the rest of lactation, we had a significant increase in milk yield. And if you add up the entire lactation, we're looking at a very similar response magnitude of what we saw with the drugs, about a 13% increase in milk yield. So that was exciting to us. The other thing that seemed to correspond pretty well with our drug studies is when we thought about mechanism, and of course these weren't really mechanistic studies on a commercial farm, but we did have milk data, including somatic cells, and we didn't necessarily anticipate this, but with both drugs that we used and the herbal extract that we used, during the first roughly 3 months of lactation, we saw a significant decrease in milk somatic cells. So this is a log transform data. So here we're looking at about a 0.5 log reduction in somatic cells with the drug treatments, and a full log reduction, more than a full log reduction here in the middle of this treatment window for the herbal extract. So this is actually, if you put it on a normal scale, it's about a 50% reduction in milk somatic cells, so pretty substantial. Okay, so we don't know really what the mechanism is here behind the milk response, but our best guess right now is that somehow these anti-inflammatory treatments, even if they're only given once in the case of Bloxacam, we think is perhaps setting up the mammary gland to be less inflammatory. So maybe it's learning to not overreact to some challenges that it's presented with, and that reduces the amount of immune cell recruitment that goes on, reduces the inflammatory tone of the mammary gland, and actually allows those cells to be more productive, hence more milk. I should point out, and I should find a way to add it to this slide, but on this study, well, in this study, we didn't see a significant change in clinical mastitis, but here we did. This herd happened to have a relatively high clinical mastitis rate. They were maybe being very sensitive in how they were diagnosing that, but regardless, they cut the clinical mastitis risk or the rate in half with this SBE 60 treatment. So that also points to the fact that we weren't just downregulating the immune system and allowing some cows, more cows, to get clinical infections. In fact, we were reducing both subclinical and clinical signs of mastitis. That doesn't necessarily answer the question of how is it even possible that let's say a drug given one time that maybe lasts for 10 days, how could it possibly influence milk yield 300 days later, right? I guess we're used to, in dairy cattle, seeing a change in peak milk, carrying out through the rest of lactation, but still, how could this work? Well, if you're not familiar with epigenetics, it's a really fascinating area that's helping us to understand how things other than the sequence of DNA can actually influence animal physiology. So epigenetics means above genetics, and it's, again, these chemical modifications to DNA, whether it's chemicals that are added to the proteins that wind up DNA, histones, or if it's actually chemicals added to the DNA backbone. These things can influence whether a gene is actually expressed or not. And it's not, doesn't necessarily mean it's totally on or totally off, but it could change, say, the expression of a milk protein gene by 10%, and you could potentially be influencing milk protein. So we wonder if this is part of the way that this targeted anti-inflammatory strategies right after calving might influence milk yield for a long time. So let me give you one neat example of this from another species. So this is a study out of Australia. In Australia, doesn't produce a whole lot of its own grain, so they're buying on the global market. And so they tend to, especially the poultry industry, tends to jump back and forth between using corn or wheat as their primary grain source for birds. And this team asked a really interesting but simple question. They said, okay, what if we have hens that are laying eggs that we're using for broilers, so that we're actually raising the chicks? What if we have a system where the hens are consuming corn and then their chicks go on to consume corn? Or what if we have a system where prices on the global market flipped, and now we've changed from feeding wheat to corn or from corn to wheat? And so they actually did this experiment, okay? And when I first saw this, I thought it was pretty astounding. So they ended up seeing it didn't really matter whether you went from corn to wheat or wheat to corn, but if you swapped grains, those chicks actually had a slower rate of gain. And by week 5, it's a noticeable difference. Now, the poultry people easily can put thousands of birds on study, so they have a lot of statistical power, and that's not a massive change, okay, but it's meaningful to them. So compared to having the offspring on the same grain as the hens were, they lost some gain. But the bigger thing is the mortality rate in this swapped treatment actually doubled. And again, it didn't matter which direction the change was, but somehow the hens had pre-programmed their offspring to be ready to eat the same grain, I guess, is one way to think about it. And if that was a surprise that they switched, it didn't help them in terms of health or growth. That's probably the best animal experiment I could show you, with a livestock example, but there's a lot of neat work going on in mice right now, kind of digging into these epigenetics questions. So that's another aspect of, we've kind of been talking about nutrition 2.0, these nutrients acting kind of like drugs and having these long-term effects even across generations potentially. And if you want to stop and think about how this might change the way we approach nutrition, I have some ideas about that. I mean, one, if you think about these things, just think about the situation of a poultry producer in Australia switching back and forth to go to the cheapest grain. Now you have to stop and think, is this hurting the next generation? Should I just swallow the slightly greater cost of this grain that I've been using as opposed to potentially setting myself up for problems? And, a lot of this is speculation still, but you can bring this to bear on the dairy industry as well. So, if we cut out feed additive X because prices are really poor right now, will we actually be influencing the next generation of heifers that we're going to raise and bring into our production herd? And there's dozens of questions we could ask along those lines. It also, I think, makes it more likely that responses vary between physiological states. So why? Well, because when an animal is, say, immediately post-calving versus peak lactation, those receptors I showed you that respond to nutrients, they can come and go, right? She can upregulate those receptors or take them away. And it's possible that those receptors changing in abundance would completely change how a nutrient acts on an animal. So it means it's really important for us to try to understand how do nutrients affect cows at specific times in their life. And then, of course, almost infinite dietary interactions that you could ask about. Like, what if one nutrient causes expression of a receptor that's necessary to respond to another nutrient? Do they have to be fed in tandem to have some benefit? And it just makes the whole system a lot more complicated. That also means that if somebody is a good salesperson, they could come in and convince you of almost anything under the sun, right? And without real data, you should be very skeptical. So, again, as somebody who sells ideas to get grants, right, I have to acknowledge this. You can come up with ideas that sound great on paper, but they may not actually show up in an animal. So research is important. All right, so I hope I haven't drugged this on too long. Just to kind of wrap this up, this is kind of our working model right now. We don't know this for certain that this is absolutely true, but there's quite a bit of evidence that fits this idea that it's probably good, or at least acceptable, for cows to have some inflammatory wave after giving birth, or right around that window of time. But in those cows that are set up to be healthy, they seem to resolve that quickly. By 3 or 5 or at least 7 days after giving birth, those markers of inflammation are kind of back to baseline. And we think that allows her to get her uterus back to normal, to her liver's functioning right. She can start eating a lot of feed, and things are going to go well. Whereas we think it's the cows that don't resolve that inflammation that are the real problem, where we get chronic inflammation in the uterus, that chronic endometritis, where we have cows that have that sort of inhibition of appetite that leads to metabolic problems, and the other things that we struggle with through peak lactation. So some of these nutritional questions, part of the part of the question we're asking is it actually going to help cows to resolve? Maybe it doesn't stop inflammation, but maybe it helps them resolve. And if we can do that, that may actually be all we need. All right. So again, if we want to approach this at the farm level, I'm not sure the situation in Israel, but here, if you wanted to do that as a farm, you could try to say every 3 or 4 months, go in and sample 20 transition cows and look at these blood markers of inflammation. And I would recommend haptoglobin because we have the most data on it. And using what we know for now, which is probably not the final story, but we at least have one cut point that we could use for now that says, you know, you should have less than 10% of cows above this high threshold of 1.5 grams per liter. Otherwise, maybe it's a problem. And the other thing I would say is, you know, all the feeding strategies I talked about, well, I guess 2 of them is all I talked about, but both of them are quite expensive. Like if you're going to feed a fair bit of omega-3s, it's going to cost a lot. The herbal extracts are generally expensive, but that's if we look at them on a cost per cow per day basis. And if some of these we can just feed for, say, 2 months or 3 months, and then calculate the cost for the entire lactation and compare that to some of the treatments we do for sick animals or some of the boluses we might give, on that scale, they don't look so bad. And because we can impact the entire lactation, it makes the return on investment look much more plausible. Okay. The other advice I would give is to monitor for changes over months, particularly if you're going to implement a new feeding strategy. I think it makes sense to go back and say, are we shifting things with this strategy or not? Are we decreasing disease rates? And certainly I want to point out that we do expect during summer for these markers to look worse. So keep that in mind when you're comparing farms. All right, that's all I have. I'd be happy to try to answer any questions you guys might have for me. Thank you.
Thank you very much, Barry, for the very interesting presentation. Okay, if someone wants to ask question, please, someone. Okay, and Uzi.
Can't hear you, Uzi.
You're on mute or something.
Now you're, oh, let's see. Nope.
Okay, later. Okay, someone else to ask?
You hear me now?
Ken.
Yeah.
Ken,
Okay. I want to ask you one question. Do you look at this high inflammatory tone around parturition, around calving, as a positive process? And if it's positive, a positive process, if you look at that, if you refer to this situation as a positive process, do you recommend to feed cows with the anti-inflammatory agent nutrients only post-calving, or we can feed them pre-calving and maybe reduce the inflammatory tone around calving?
Yeah, good question, Uzi. And this has been discussed a lot here. We need more data, I would say. But what my speculation is, even though we probably need to allow some inflammation, I don't think it's likely that any dietary anti-inflammatory strategies are so potent that they're going to cause a problem with calving. I could be wrong. But I think the system is able to make, say, sufficient prostaglandins, even if you have a reasonable amount of omega-3s around, or if you have these flavonoids from plant extracts around, regardless, because they're nowhere near as potent as like flanoxin at suppressing cyclooxygenase enzymes. And in fact, I showed you the one study we did with that Chinese skullcap extract, and I've been kind of advising a company that's now marketing that here, and they've done a number of studies where it's been fed before calving and after calving, or just after calving. There hasn't been any obvious problems with feeding it before calving, and it's mixed. I mean, research data is always noisy, right? But it looks like on average, the cows at least are more productive if you also feed it pre-calving. And that would be my bias. The only reason we didn't do it in this one study is we used the robots to deliver the treatment. We couldn't come up with a logistical way to dose the cows pre-calving on that study. Does that answer your question?
Okay.
I want to ask you, we're talking about, we're talking about cows, then the best treatment is to prevent them from being sick. And in this regard, I want to ask, if we talk about treat the cows during the dry period or before calving, did you look on treatment just to specific cows according to different parameters, like cows with high body condition score during dry off, cows with low milk production, and that they are more, have more chances to be with metabolic disease or metritis after calving? Do you think about?
Great question. So far, we have not. The strategy we've used is to try to balance treatments by those risk factors. Body condition is an example. When we've done these larger studies, we've tried to have a balance of body condition across treatments. And then we've later asked, statistically, can we show evidence that the benefits of the treatment are greater, right, in the high body condition score? So we've tried to use that sort of approach to ask if we can find a subgroup that's maybe going to benefit more. And, we haven't done a 1,000 cow study to date, so to some extent we haven't maybe had enough animals to do that properly, but we haven't so far succeeded at finding a predictor. Even haptoglobin hasn't helped us predict which cows would, show a greater response. So we want to get to that, but I've consulted with some drug companies trying to go down this path. The problem is, if you do need to treat quickly, and that's been my assumption, which may be wrong, that we want to treat in the first 2 or 3 days. If you're going to do that, whatever indicator you're going to use, you have to get an answer pretty quickly, ideally right away. So we've had a lack of tools to measure, say, haptoglobin cow side, and there are people working on that, but we don't have anything right now. And I've spent a bunch of time digging into, could you use like body temperature? And it's very difficult to draw a cut point because it changes so much with season. And body condition score would be obviously an easy one, but we have no evidence so far that treating just the fatter cows would help them more. But it's a testable question that, one of many things that come to my mind and I would like to follow up on at some point. It's a good question.
יניב, אני יכול שאלה?
כן, בבקשה.
Hello, first of all, I hope that first of all, it was a great lecture, very interesting. And I hope my question won't embarrass my professor, Moalem, which was the first question. So I hope I will get a good one and not put any shame, on him. So basically, I'm a field nutritionist. Me and some of my co-workers here, we work for the Extension Service in the Ministry of Agriculture. And like you said, you talked a lot about strategies. And my question is, I know it's a little bit of a hard question because sometimes I look at it as more of like the end point. So if you would use one product, because we feed usually one ration to all lactating cows, postpartum and high lactating, and everybody gets pretty much the same ration, and which is very hard to separate. Even if you want to just go with postpartum cows, it's really difficult. So I know some of the products, and I as well in my thesis did on omega-3. Is there a product that can benefit maybe the entire lactating group, or something that is more common that you would recognize that can help this postpartum period? Thanks.
Yeah, that's a great question. We have varying degrees of that same issue here, but I think it's a bigger challenge for you. Well, I don't think, and this is where I would disagree with the Florida group a little bit. For example, I would be totally fine feeding some additional omega-3s, the whole lactation. The problem really is the cost, not that it's not going to help all the cows. And that's a major problem. But there are more and more of these polyphenol sources. Many of them are flavonoids, so I'll just say flavonoids. More and more products. I don't know about Israel. I know there's way more in Europe than there are in the US. And I think many of them don't have data, but they're at least feasible that they could help, and I probably wouldn't have a problem. The one we tested in the US costs something like 16 cents per cow per day, which is huge. Like it's a problem to convince people even to feed that for 3 months, much less 12 months of the year. So I don't know if there are cheaper options like that, even if the evidence isn't really there, where it's maybe 2 or 3 cents, and you maybe take some risk that it's giving a benefit, even if you don't have direct evidence that it is. But I don't know if that's available there. Can you tell me what?
Yeah, it is available. We have some phenols products. I think oregano. I don't know. It's oil that now I don't know if the company that sells it are here, and when they heard you, maybe their eyes popped out hearing you recommending phenols, and now some of them are like running into, and maybe we'll get some letters. But there are some products here, and if it's okay, I would love to send you some of them, and then you can check it out if you think it's suitable to the ones you talk about.
I would certainly like to look at the details. Yeah. Well, I will say one thing that's shocking about this study we did, those cows only got 10 grams per day of this extract. And of course, the extract isn't 100% flavonoid. So if you do it per body weight and on a huge cow, if you would have told me, like before I saw any of the data from France, that you would feed that much flavonoids and see any kind of response, I would probably have laughed at you. I mean, I was very skeptical. I thought we would do this study and be like, yeah, I didn't think it would show anything, but so I don't know. There's a lot to learn still. But
thank you, and thank you for saying it's a great question. Now I can, go to sleep feeling well. I had a good question, not putting any shame.
Uzi is proud.
Yeah, I hope.
We talked about it tomorrow morning. Yeah.
I'm waiting for the call.
Yaniv, can I ask a question?
Yes, of course.
Hi, Bear. I wanted to ask, there are many supplements, feed supplements that change or things in the rumen, like yeast supplements and others. How do you think, if at all, they could affect the inflammatory processes postpartum?
yeah, well, you might know this, Maya, but we've done maybe 3 studies like that now with yeast products, where we've specifically looked for if we see changes in immune measures, as well as inflammatory measures. And actually, at least one of those products, I've now seen, I think, 3 studies where we see an increase in haptoglobin with the yeast product. So if anything, it's making things more pro-inflammatory. Now, it also tends to improve animal performance, at least a little bit. So I think when you do a treatment, just because on average cows with more haptoglobin have less milk yield, doesn't mean a treatment that raises haptoglobin is necessarily going to decrease milk yield or cause things to be worse, right? So things get complicated there. But I haven't seen any evidence that a yeast product actually decreases net inflammation. Now, there's nice studies that show that yeast cells can bind to pathogens in the gut and cause them to flush out into manure and maybe prevent disease. And I think, I wouldn't say that this is rock solid, but from the literature that I've seen with all these yeast studies, my general take is it's not a bad insurance policy, where if a cow gets challenged with, say, mycotoxins and some pathogens and that sort of thing, I think on average they help a cow deal with those challenges better. And then by doing that, she would get less inflamed because she's not getting sick. that's my best. If I was a nutritionist, that's the way I would approach it, you know, in the field, is this is the value of this, as opposed to making every cow less inflamed or healthier. Do you agree? What do you think?
No, I wanted like specifically to ask you about what, you know, there was a lot of talk in the last decade about microbiota and their role in the physiology. So, and that's what I wanted to ask you, if you think that changing something with the rumen function, the microbiota there could have systemic effects in the cow.
Well, I guess, first of all, I'm not a huge like microbiologist, okay, but I'm a little skeptical that changing the rumen microbiota is likely to have much effect. I think hindgut microbiota, which is probably more directly related to inflammation in the system, because you have one layer of epithelium instead of 4, and more likelihood of things interacting with the immune system, because there's not a lot of immune cells at the rumen surface. The kind of counterintuitive thing that I think about in transition cows for the rumen is there's some neat studies on dietary sugar at like 5 or 6% of dry matter improving transition to lactation. Not every study has shown that, but some have. And I think it's partly by improving blood flow to the rumen, and thereby improving, well, sorry, it gets fermented to butyrate. Butyrate is very good at driving epithelial growth in the rumen and blood flow to the rumen, and that helps you get quicker uptake of BFA and prevent accumulation of BFA, which Jorg Aschenbach's group in Germany did a really neat study showing too much BFA, even at a regular pH, can cause disruption of the rumen epithelium. And then you get endotoxin coming through the rumen and other problems. So I guess sugar is, to me, the tool that I think of in that respect.
Okay, someone want to ask more questions? People are still here, or everybody left us?
Some people are awake at least.
Yes. Okay. Yes. I want to ask, if there's no more question, one question. Do you think that if we have now going to start a new project of trying to find different parameters in milk samples, and the idea at the end is to try to find those parameters in milk during the milking online. And we thought about different parameters, and now after you spoke, I thought that if we can find a parameter for stress, maybe haptoglobin, maybe cortisol, maybe something else, and then to find the stress of cows according to these parameters and to treat those cows, do you think it will be something that will be helpful?
Yeah, I like the idea. I will share that, you know, some of these big farm studies, we really wanted to be able to use milk samples if we could for assessing inflammation, because, you know, bleeding cows on these commercial farms is not super easy. But we kind of gave up on that because most of the measures that we've used, or almost any inflammatory measure that we thought about, if you measure it in milk, it probably came from the mammary gland. So it's basically just another way to get at mastitis. It doesn't really tell you a lot about the whole body's inflammatory status. So maybe that still has value for what you're wanting to do, but I don't think milk, haptoglobin, and plasma would line up very well. Now, cortisol, to my understanding, wouldn't really be made in the mammary gland. And so maybe that's a better like sample of whole body cortisol, and maybe that makes some sense to me. There's probably other things like that.
The plasma.
I think,
Yes.
I think there will be another problem, even if you'll identify stress cows, of how you'll treat them individually. So what, by injection? If it's by feeding, so all the group will be fed with this, so, you know.
Yes.
How do you treat them?
By injection, yes, individually.
Ah, that's okay.
Like we talk about individual, yes, but now use farms. Nobody wants to look on the one specific cow. This is one of the problems, I think.
Yaniv, this is my fault for talking too long, but I kind of didn't realize.
No, it's okay.
I have a meeting. I have a meeting starting now.
Yes, okay. Thank you very
My
much.
fault.
Yes. Okay, thank you very much
Bye-bye.
for joining us.
Bye. Thank you. Thank you.
Thank you.
Thank you.
Yeah, I
Bye-bye.
enjoyed it, guys. Thanks for the
Yes.
opportunity.
And good evening, everybody.
Thank you. Good evening.
Thank
Take
you,
care.
everybody that stay until now.
Thank you.
Okay, goodbye.
Bye-bye.
Bye.